Hello everybody! In this video you will learn more about our new bacteria Extraflore Pure Fruit. To start this presentation, I would like to say first that we based our selection work on the study from the lab of Mr. Patrick Lucas from ISVV in Bordeaux. He worked on the lactic bacteria biodiversity. He made several researches on the geographic origin of various strains of Enococcus uni. The main goal was to determine if there are geographic specificities and if these strains have special capacities for malolactic fermentation. You probably recognize this kind of figure that shows a genetic proximity between several bacteria strains. This genetic tree was proposed by El-Khoury in 2014. He analyzed 43 lactic bacteria strains and more especially the single nucleotide polymorphism. SNP. We clearly see two main groups of bacteria in this study. The group A, composed by 21 strains, is genetically different or far from the bacteria of the group B. There is also a third group, N, that is between A and B. Here on this slide, El-Khoury proposed another view more detailed. This time, he analyzed 577 strains by SNP method. Each circle represents a group of bacteria genetically similar. The various colors in each circle show the geographic origin. Regarding this graphic, we can say that one strain of bacteria could be found in many various wine regions. For example, the group A2, the biggest circle, is present in Bordeaux, but also Languedoc-Roussillon, Lebanon or Burgundy. It's the same for the group A1, the bottom of the graphic, that is also present in Loire Valley. We can say that in general one bacteria strain is not representative of a terroir or specific from a region, except if we focus on the group A5 and A28 that is mostly composed by bacteria from Burgundy. It seems that these both groups are clearly different from others. Please also note that the group N and B are mainly composed by specific bacteria from cider. Just before, we could see that bacteria strains are not linked to a geographic origin except some strains from Burgundy. So El-Khoury decided to observe if bacteria are more or less linked to a matrix of wine, white, rosé or red. And this time again, in the group A1, composed by 116 strains genetically close, and A2, 148 strains, he found indifferently these bacteria in red wine, white and rosé. Except one more time for the group A5 and A28 that are respectively composed by bacteria coming from Burgundy white wine for the group A5 and Burgundy red wine for the group A28. So, for a new bacteria selection, we decided to focus more on the bacteria from Burgundy group and wanted to see if they have specific capacity to succeed the malolactic fermentation, and especially in hard conditions. IOC is aware that many winemakers are concerned by that, and a high level of alcohol is one of these conditions. So, before continuing our selection, we would like to be sure that the bacteria from Burgundy are resistant to high alcohol concentration. We could think that it won't be the case, but the study from El-Khoury showed us that the Burgundy strain has very good capacities to survive in a matrix containing 16% alcohol. So IOC decided to continue the selection. Moreover, it's funny to select a bacteria in a cool climate region that will be adapted for meridional vineyards. The selection project was initiated by Comité Champagne and IOC. We selected a new lactic bacteria in a red wine and tested it in several conditions during 3 years. So, for a new bacteria, we decided to focus more on the bacteria from Burgundy and wanted to see if they have specific capacity to succeed the malolactic fermentation. We tested it in various conditions and pay attention on the following parameters: temperature, alcohol, initial concentration of malic acid, and pH. I let you discover now some results. We observed first the kinetic of fermentation of 4 various bacteria on a red wine at 20 degrees. We could read on the graphic that there is no difference between the 4 strains. But at 16 degrees, we could observe some differences between the strains of bacteria. On the screen, we compare Extraflore Pure Fruit with another bacteria, respectively the green and blue line. We see that in the both cases, the malolactic fermentation is running, but Extraflore Pure Fruit finished the fermentation 10 days before the other strain. The kinetics are similar when we double the dose of bacteria. We also made some tests on rosé wine at 16 degrees Celsius, and Extraflore Pure Fruit confirms its good capacity to succeed the malolactic fermentation. On this matrix, its kinetic is close to maxiflor that is recognized as being resistant in our range. As we saw it previously, we based our selection on the alcohol resistance. On this slide, we tested 3 bacteria in a blend of Grenache and Syrah at 16.8% alcohol. We can see that Extraflore Pure Fruit started more quickly the malolactic fermentation and finished it 1 month before the 2 other strains. We also know that a low concentration of malic acid is often a problem to start the malolactic fermentation, and it's a current situation, especially in meridional vineyards. The graphic on the screen shows a synthesis of various dries made on Merlot, Sangiovese or Grenache. All the wines had less than 1.2 grams per liter of malic acid. In purple, we can see that Extraflore Pure Fruit always achieved the malolactic fermentation earlier than the control. And it's mostly due to the fact that the strain started really quickly the malolactic fermentation. And now the pH. Even if it's less a restrictive parameter for meridional region, we controlled this parameter. At pH 3.2, we can see that Extraflore Pure Fruit finished the malolactic fermentation after 10 days. We also observed in gray that a spontaneous fermentation started in the wine after 13 days. But when we made the same experience at pH 3, the spontaneous bacteria never started. The bacteria Ref3 and Extraflore Pure Fruit succeed both the malolactic fermentation, but Extraflore Pure Fruit started it faster and the delay was shorter. Another important point for us was that the selected strain should not produce diacetyl. We measured the diacetyl concentration in several modalities, and each time with Extraflore Pure Fruit we obtained less butter aromas in the wine. And the fact that Extraflore Pure Fruit produces no diacetyl means that wines are described more complex with fresh fruits aromas. Several tastings confirmed it. So to conclude this video, we hope that Extraflore Pure Fruit will answer to your requests, especially if you expect quick malolactic fermentation and or if you have difficult conditions as low temperature or high alcohol. I hope that you enjoyed this video. Bye bye!